Yes, the next lecture will be given by Gudrun Fema Olafsdottir, who was a student at Reykjavik University but is now working at Iiim. So it's an example of a project that has migrated over to the Institute for Intelligent Machines. And it really is sort of showing us how the power of simulation can teach us something about very complex things, such as life itself. So I'm very excited about presenting Gudrun to talk about that in more detail.
Okay, so my name is Gudrun Fema Olafsdottir, and today I'm going to talk about simulated self-directed growth of human tissue. This is a project I've been working on with Chris for a few years now. So in the beginning of this research, we started with the question, how does a cell turn into a body? Or how does the body know how to become a body? And if we could possibly answer this question, we could have like great benefits of it, both to medicine and to scientific understanding of biological processes. So we could possibly prevent or detect birth defects, or before even when it's a fetus, not when the baby is already born. So we started with the smallest thing. We started with veins. Everything about veins is known. So we know we know all the cell signals, we know the differential equations, we know the physical terms, we know everything about it. So, and already today the signals have been written, and people have created simulations of the morphogenesis of veins, and those are some of the prior work that has been done in this field. And yeah, so if we conclude, so what has been done is the cell signals have been written, and we have created simulations. But the simulations have been created by using partial differential equations. The differential equations are the most common way to simulate biological systems today and other multidimensional systems. But they are, of course, continuous, and they are kind of obvious. If you change the parameter, you can predict the outcome. You can see what effect it will have. And it also elucidates complex processes in compact format. But it requires physical terms in details. So if we don't know the physical terms in details, like the kinetic laws or something like that, we can't really simulate the processes or the morphogenesis. So in my thesis work here at Reykjavik University, I introduced a new methodology to simulate tissue growth. And it is really complementary to prior methods. It is based on cell signals like the others, but it doesn't require partial differential equations. So the question we asked ourselves at the beginning was, how do we simulate the growth of veins, which is called angiogenesis. So what we do is we go into the literature and find the cell signals. The next step is to create a process algebra model, which will then be translated into a finite state machine. And from the finite state machine, we will create cellular automata rules. So the CA rules are grounded in the literature. So we know that the cellular automaton model will actually represent the actual cell signals. So what we need to simulate in angiogenesis is the branching of veins. So when smaller veins are growing from like a bigger one, and also just the growth of veins. So this is an image of all the cell signals that occur in angiogenesis. And what we do is we take the signals and group them into specific categories. Autocatalysis, which is actually the effect that the signal has on it. If a cell sends a molecule into the extracellular fluid, it will have some effect on the same cell that sent the message. So it's actually sending message to itself. Then we have positive feedback, which is an activating molecule that goes to another cell and triggers a positive. Then that cell will also send out a positive molecule. Then we have negative feedback, which is inhibiting. Then we have the diffusion, which is essentially the travel of molecules in the fluid outside of the cell. And it is a message to another cell. The decay of the molecules that are diffusing in the fluid outside the cell. And then the differentiation, that is when the cell split into two. So after analyzing the signals into these groups, we created a Bio-PEPA model, which is a process algebra model. And this process algebra has been developed in the last 8 years. And it is used to define a system that starts from the definition of its subcomponents. And this is how we can create complex systems just from with only the information that the cell has, not the system as a whole. So it offers a high level of abstraction and with a focus on the subcomponents and the interactions. So in our model, the subcomponents are the cell, and the interaction are just the messages that they send, either to just the cell that is adjacent or the cell that is far away through a molecule. Okay, so here is a Bio-PEPA model of the angiogenesis. It's quite simple, like this. And this is just an intermediate step. The Bio-PEPA can be used as an intermediate for different kind of analysis and modeling. So, and one of the steps or one of the analysis we can do is we can translate it into finite state machine, as we do here. The green arrows are the information that we add after we have created the Bio-PEPA model. It cannot be represented in the model. So that is diffusion and also inhibiting molecules that are sent out to the extracellular fluid. So from the finite state machine, we derive cellular automaton rules, and we've just grouped them here. We have 2 rules for autocatalysis, 2 rules for decay, and 1 for inhibitor, 1 for differentiation, and 1 for diffusion. So the CA model will look like this, something like this. So here we see a simulation of a growth of a vein. This is just the starting of a vein. So we can see that it actually grows, and it knows how to stop, it knows how to begin, and yeah, it is at time 55, 75, and 100. So we can see it grows very quickly after it has started. Here we have another example of a cellular automaton model, which shows us both the growth and the branching. And so, okay, so we have created the model, and then we asked ourselves, how can we know that these rules are actually what happens in the morphogenesis? So, as I said before, everything in angiogenesis has been created or is known. It has been done before. The simulation has been done before, just with another methodology and with another approach. So for example, this Göre and Kurz, they did this. They did a simulation of the angiogenesis by using partial differential equations. And what they did is they took the basic physical terms, and then they were lacking some few others. So they did some studies and found out the partial differential equations for the other physical terms. They created probability functions and partial differential equations. They iterated this process, and at the end created a successful simulation of the morphogenesis. So here on the left is a picture of or image of their model, and on the right hand we have our model. And again, here's another simulation. And then we compare them with real life. So if we compare the branching with real life, we can see that it's actually quite similar. And with the whole picture, so we have a real tissue here at the bottom, and then at the left, Kurt's and Gare's model, and then our. So what our results are is a high level of visual similarity achieved. So based on actual cell signs, so or signals. So what we know is that we can actually ground them in the literature, and they actually represent real life, what happens in real life. And the rules are generated by the new methodology, and without any need of differential equations, which is a major advance because we don't know the kinetic terms for most other tissues. So this is a promising line of inquiry to speed up modeling of complex biological processes. And as I said, this methodology applies to almost everything, almost all biological processes. So, and as we can see, with this, we haven't simulated any of this before. So we could apply this methodology to anything, anything up there, and we could hopefully successfully create a model of the growth of these tissue. So the next steps is to develop new math for complex systems. We want to create like something, so we don't have to create the Bio-PEPA model and then create the finite state machine and add some information. We just want a new math that has everything in it. So, and then we want to produce the differential equations from the cellular automata model and close the circle. So we can actually create this model with it is an abstraction. So we will create the cellular automata model, which actually represents the cell signals, and from the cell signals we can create the differential equations. So we can predict everything that is lacking in the literature today, like the physics terms. So yeah, it's a major advance. So thank you.
Good. It's time for questions. I'm curious to know, actually, so is this a tool that you will be giving out to people to try, like a modeling system that you will then start using today, or something?
Maybe not today, but hopefully in the future, yes.
Mm-hmm. And you've received some interest in doing it this way?
Yeah, it's normally in biology is that you can't simulate the processes if you don't know the physical terms, if you don't know the kinetic terms. But here we can actually predict and say what the physical terms are, so that we are lacking. So, yeah.
Mm-hmm. Exciting. Any other questions?
Yeah, is there some other way of evaluating if the model is, except just look at visual?
Yeah. Yeah. So as I said, the next step is to actually derive the differential equations from the model, from the cellular automaton model, and if we can, and actually the differential equations do exist today for like morphogenesis of veins. So if we find the differential equations from the cellular automaton model and compare it to the real.
It's not specific to what you're doing, it's just also for the previous work. Like, so they were showing us a picture. So how did they evaluate the appropriateness of what they did? Like, or is there any other way? It might not be that just looking at it visually.
Today, it's not possible, except unless you have all the kinetic terms, unless you have like all the mathematicals.
Yeah, but even though you have that and you want to know it's correct, how did they know that it's correct? So they had to look at it just visually. But that was what
Yeah.
what I was thinking about. Is there any better way than just looking visually and say, okay, it looks alike? I don't know. It's just, and again, it's just nothing specific to what you.
No, I understand. I understand. It's the problem we had at the beginning. So, in the beginning of, well, at least at first, we will look at it visually, and if it, yeah, and actually we trying to convince ourselves that because we take it straight from the literature. So if it's actually doing the same, and we take it from the literature, it should be quite close to it. So what is the next step is to actually find also the way to ground or to confirm it that it's actually right, except for just for visual.
Just to follow up on that, I guess the question may be stated as follows. How much work is it to find these physical terms? Which is, ultimately the way to verify it, because then you have essentially made a list of all the physics, right?
Yeah.
Yeah. How much work is that for, say, for the veins?
It's known for the veins.
No, I mean, yes, it wasn't known. How much work was it from wasn't known to known?
I think it's, I'm not really sure if I can answer, I can't answer it like, but but it's a quite simple process, and it has 9 rules, so it's not really difficult, and we don't have like many differential equations, so I'm not really sure how long it will take. It's
That's certainly longer than your method.
yes, that's true. Okay.
Do you have, what would be the next thing you would want to simulate?
Okay, the first thing I wanted to simulate was the heart, and that's of course a quite big step from this. Hopefully, after this, when I finish this, I will go to the heart and start looking at the first stages of the heart development. So, yeah, but maybe it would be better to do something else. But I think that because there are not many, the morphogenesis of many muscle or biological tissue is not known. So, why not the heart? Okay.
Thanks again.
Thank you.